Hi guys, welcome back to my YouTube channel Biology with Huria. Today we are talking about pilocytic astrocytoma. Pilocytic astrocytoma are relatively benign tumor that typically affect children and young adults, most commonly located in cerebellum. They also may involve the third ventricle or optic pathways, the spinal cord, and occasionally the cerebral hemisphere. There is often a cyst associated with tumor and symptoms appearing after incomplete resection of lesion may be associated with cyst enlargement rather than growth of solid component. Tumors that involve the hypothalamus are especially problematic because they cannot be resected completely. A high proportion of pilocytic astrocytoma have activating mutation or translocation involving gene encoding the serine threonine kinase BRAF, which result in activation of MAPK signaling pathway. Pilocytic astrocytoma do not have mutation in IDH1 and IDH2, supporting their distinction from low-grade diffuse glioma. A pilocytic astrocytoma often is cystic with a mural nodule in wall of cyst. If solid, it is usually well circumscribed. The tumor is composed of bipolar cell with long hair-like processes that are GFAP positive. So xanthal fibers, eosinophilic granular bodies, and microcysts are often present while necrosis and mitosis are rare. Ependymomas often arise next to ependyma lined ventricular system, including central canal of spinal cord. In first 2 decades of life, they typically occur near the fourth ventricle and constitute 5% to 10% of primary brain tumor in this age group. The adult spinal cord is their most common location. Tumors in this site are particularly frequent in the setting of neurofibromatosis type 2. The clinical outcome for completely resected supratentorial and spinal ependymomas is better than for those in posterior fossa. In the fourth ventricle, ependymomas typically are solid or papillary masses extending from ventricular floor. The tumors are composed of cells with regular round or oval nuclei and abundant granular chromatin. Between nuclei is variably dense fibrillary background. Tumor cells may form round or elongated structures that resemble the embryological ependymal canal with long delicate processes extending into lumen. More frequently present are perivascular pseudotorsates, in which tumor cells are arranged around vessel with an intervening zone containing thin ependymal processes. Anaplastic ependymomas show increased cell density, high mitotic rate, necrosis, microvascular proliferation, and less evident epidermal differentiation. Basically, all these are important as well. Now we are talking about midline gliomas that are infiltrative and result in significant neurological impairment because of disruption of critical nearby structure. Although they may not show typical high-grade features such as necrosis or vascular proliferation, they often behave aggressively. The lesions typically have acquired point mutation in histone H3, consequences of which is loss of lysine residue that is target of post-translational modification that regulate gene expression. Another example of oncogenesis via alteration of cancer cell epigenome. Precisely how this mutation contribute to cellular transformation remain to be determined. You can ask me anything in comment section and must subscribe my YouTube channel Biology with Huria.